This is Lecture 7 on Photosynthesis, the Carbon Reactions, Part 2. This is part of the subject Plant Physiology, which is offered at NMIT as part of the agricultural and viticultural degrees. Would you please refer to Chapter 8, Tays and Zyger, for complementary readings. My name's Dr. Nikki Cooley. It is important that you not only watch Part 1, Photosynthesis, the Carbon Reactions, but that you also understand that the Kelvin-Benson cycle and that these have 3 phases. If you're having trouble understanding these phases, the following YouTube video may help you in understanding these components. Please note though, in this video they do describe carbon accounting differently to how we have. They multiply the inputs by 2 so that all the inputs add up to 1 molecule of glucose. In our example, we counted based on the production of 1 molecule of PGAL. Both methods are commonly used and both methods are correct. In Part 2, we are going to cover the following topics. We're going to look at a little bit more detail about the enzyme rubisco. We are also going to introduce another type of plants called the C4 plants and the Kray's anatomy. We are going to follow by an introduction to the CAN plants. So let us review for a moment. Rubisco is an important enzyme used in the regeneration of the Kelvin-Benson cycle, and this was covered in Part 1. The role of this enzyme is to catalyze RUBP. This is an essential part of the regeneration phase. Sometimes this does not always happen in plants, and there are problems in the regeneration phase. This problem with rubisco is a very important problem in agriculture, and so we are going to spend a little bit of time trying to understand this. In order to understand the problems with rubisco, it is important that you understand a little bit about evolution. Plants and the Kelvin-Benson cycle evolved within the Earth's atmosphere at a time when there was no oxygen present. It turns out that rubisco can bind more readily to oxygen than it does to carbon dioxide if both are in the facility. So why is this a problem and how does it occur? So let's start with why oxygen should occur. If you remember in the first lecture, we were told that CO2 comes from the atmosphere, and it can come into the plant via the stomata. It then dissolves through the cell into the stroma, where it can undergo the Kelvin-Benson cycle. When stomata are open, this process occurs well. However, what happens when stomata are closed? Well, as you remember, oxygen is a byproduct of some of the reactions. This oxygen builds up and then becomes present in the stroma. It is the build-up of this oxygen which then enables it to compete with the carbon dioxide, and as rubisco binds more readily to oxygen than it does to carbon dioxide, it means that there are inefficiencies in this system. So let us review this reaction. If stomata are open, an atmospheric CO2 can enter the leaves and go into the stroma. Carboxylation yields 2 molecules of the 3-phosphoglycerate. However, if the stomata are closed and a build-up of oxygen occurs in the stroma, different compounds are yielded. 1 molecule of phosphoglycerate is yielded, as well as 1 molecule of 2-phosphoglycerate. This is not used in the Kelvin-Benson cycle. Oxygenation involves reactions in the chloroplast, leaf peroxisomes and mitochondria. This process is also known as photorespiration, and it causes partial loss of the production of on the fixation of carbon dioxide and the uptake of oxygen. These competing reactions have negative impacts on plant growth, and these also have negative impacts on productivity, yield, and can be seen often in agriculture, especially agriculture occurring in Australia. So the CO2 and the oxygen compete for rubisco with RUBP. This is known as the carbon-2 oxidative photosynthesis. Oxygen creates 2-phosphoglycolate and 3-phosphoglycerate. 2-phosphoglycate is then metabolized by two other organelles, the perosomes and the mitochondria. In the recommended textbook, Figure 8.8 describes the process about what happens in these two reactions. So let us review. The balance between the Calvin-Benson cycle and the C2 cycle is determined by three factors. The first is inherent to the plant. The second is temperature, usually higher temperature, causing a tendency towards the C2 cycle rather than the Calvin-Benson cycle. And thirdly, the concentrations of the substrates, carbon dioxide and oxygen, in the stroma. If we look at increases in temperature, this modifies the kinetic constraints of rubisco, increasing the rate of oxygenation. Temperature also lowers the concentration of carbon dioxide, more than that of oxygen in a solution in equilibrium with air. So you can begin to see that a progressive increase in temperature tilts the balance away from the Calvin-Benson cycle towards the C2 oxidative photosynthetic cycle. The adaptive value of photorespiration is a matter of debate. Firstly, it is thought to promote the recovery of carbon that would otherwise be lost as a 2-phosphoglycolate. Secondly, the formation of 3-phosphoglycerate prevents the lethal accumulation of glycerate in dead-end pathways. Thirdly, thought to minimize photoinhibition caused by excess reductant formed in chloroplasts under stress, for example high illumination, high temperature and carbon dioxide or water deficit. And lastly, the major source of hydrogen peroxide is photorespiration. Although toxic, hydrogen peroxide also acts as a major signaling molecule and regulates the cell homeostasis, and thus has a positive role. Let us talk about engineering of photorespiration. Efficient ways to increase biomass are being sought as solutions to the global food and energy shortages. Can land plants be adapted to enhance photosynthesis? Well, enhancing photosynthetic efficiency or reducing photorespiration should improve yields of crops such as wheat, rice and cotton. Attempts to modify the performance of rubisco, unsuccessful to date. Plants are generally stunted and die under atmospheric CO2. Alternative pathways for retrieving carbon of 2-phosphoglycolate has been introduced into chloroplasts. These are via bacteria E. coli, glycocarbotic pathways. This lowers the flux of metabolites through peroxamides and mitochondria, thus avoiding the use of ATP. To date in our lectures on photosynthesis, the carbon reaction, and on photosynthesis, the light reaction, we have only explored one type of carbon metabolism process. The plants that exhibit this kind of metabolism are described as the C3 plants. But what if there was another way? In the next part of this lecture, we're going to explore some other types of metabolism. C4 plants have evolved anatomically to be different from C3 plants. These differences allow the plant to compartmentalize the carbon fixation part of photosynthesis. Why do this? Well, to minimize the oxygenase activity of rubisco and the loss of carbon through photorespiration. C4 photosynthesis evolved to compensate for limitations to low carbon dioxide. In the 1950s, C4 photosynthesis was discovered in sugarcane and maize. First stable products are the 4-carbon malate and aspartate. It takes place in two cell types called mesophyll and bundle sheath cells. The enzyme phosphophenylpyruvate carboxylase, or PPKs for short, catalyzes the carboxylation rather than ribisco. The metabolism in the C4 plants, such as sugarcane, is called the C4 carbon cycle. The resulting 4-carbon acid flows across the diffusion barrier to the vascular region and is decarboxylated, releasing carbon dioxide. This carbon dioxide is refixed by ribisco via the Calvin-Benson cycle. The C4 cycle is associated with a particular leaf anatomy, and this is called the Krebs anatomy. An inner ring of the bundle sheath cells around the vascular tissue and an outer layer of mesophyll in close contact with the epidermis describes this anatomy. This anatomy enables the compartmentalization of enzymes for C4 in two different types of cells. The image on the slide shows a C4 plant's anatomy. It is this inner ring, demonstrated by the mouse here, that shows the bundle sheath cells around the vascular tissues, and an outer layer or mesophyll cell which is in close contact with the epidermis. So, let's look at this in a little bit more detail. Carbon dioxide is fixed twice. Firstly in the mesophyll cells, and secondly in the bundle sheath cells. In doing this, the C4 pathway keeps oxygen away from RUBP. C4 plants have evolved to use a compound called PEP, and this is used to fix carbon dioxide. The evolutionary advantage of the development of PEP is that it does not bind to oxygen, unlike RUBP. So in these plants, oxygen is no longer a problem. So therefore, the fixation of hydrogen carbonate through carboxylation of PEP by PEPKs in the mesophyll cells. This converts the substance into oxyciliate. This enables the conversion of malate or aspiliate. The second stage is the transport of the 4-carbon malate to the bundle sheath cells. Remember, the first part occurred in the mesophyll cells. The next stage is decarboxylation of malic into carbon dioxide and pyruvate. The carbon dioxide enters the chloroplast and then the Calvin-Benson cycle. In the fourth stage, the 3-carbon pyruvate is transported back to the mesophyll. In the final stage of the CO2 journey, pyruvate is converted back to phosphophenylpyruvate. This is also described as the regeneration of the bicarbonate acceptor. The formula below at the bottom of the slide shows the C4 metabolism process. Pyruvate plus inorganic phosphate plus ATP gives phosphophenylpyruvate plus NADP plus inorganic phosphates. The name C4 comes from the first intermediate of the cycle, oxalate. Oxalate has 4 carbons, and hence the name. So to summarize, 2 cells are involved in one metabolic pathway. It might be easier to demonstrate this pathway and the description above with an illustration that shows both the metabolic processes and the role of the anatomy. Hopefully this illustration will make the C4 pathway clearer. So carbon dioxide from the atmosphere can dissolve into the mesophyll cells. Oxygen also dissolves into these cells, but as the compound PEP is used rather than RuBPH, all the oxygen is ignored and only the CO2 is picked up or utilised in the cycle. A C4 compound is produced, which then moves the CO2 into the bundle sheath. Once the compound with the CO2 is in the bundle sheath, this carbon dioxide can then enter the Calvin-Benson cycle, which is the same as C3 plants. The intermediate 3PG is released from this cycle and sugar is produced in the vascular tissue. You will notice that both of these pathways are cyclic. The pathway in the mesophyll cells allows regeneration, and the Calvin-Benson cycle allows regeneration. If you are struggling, then please refer to the following video for further details. This video can be found on YouTube under the following link, and it describes both the C4 and CAM plant metabolism. The C4 cycle is known to occur in 18 families of monocots and dicots, particularly in the Poaceae. That includes corn, millet, sorghum and sugarcane. The transport processes facilitated by the plasma desmata generates a much higher concentration of carbon dioxide in the bundle sheath cells, thus suppressing oxygen and photorespiration. And these are the advantages of the development of this system. The bundle sheath and the mesophyll cells show large differences in enzyme composition. For example, PEPKs in the mesophyll, ribisco in the bundle sheath, NADP malic enzyme in the mitochondria, and PEPKAs in the cytosol. Starch accumulates mainly in the chloroplasts of the bundle sheath. So let's talk about the C4 cycle and temperature. Elevated temperature decreases both the capacity of ribisco and the solubility of carbon dioxide. In C4 plants, two features reduce these effects. The first feature is a high affinity of PEPAs for its substrate saturates the enzyme as a reduced carbon dioxide concentrations. This in turn enables C4 plants to reduce stomatal aperture, conserving water while fixing carbon dioxide at rates greater than C3 plants. The high concentration of carbon dioxide in the bundle sheath cells minimizes C2 photorespiration, both significant advantages. It is the anatomical features that minimize water loss in CAM plants that include thick cuticles, large vacuoles, and stomata with small apertures. This allows tight packing of mesophyll cells. Typically, a CAM plant loses 50 to 100 grams of water for every gram of carbon dioxide it gains. This is approximately 250 to 300 grams for the C4 plants, and 400 to 500 grams for the C3 plant. So these plants, the CAM plants, keep their stomata closed during the day, all the time. This is different from C3 and C4 plants, as while they will close their stomata during the day, it is usually only in response to a specific climate condition at that time. When CAM plants have their stomata closed during the day, they are able to reduce their water loss significantly. But this limits the amount of carbon dioxide that they can obtain from the stomata and use in the photosynthetic processes. During the night, cam plants have evolved to be able to overcome this problem by collecting CO2. The CAM4 plants fix carbon dioxide by PAP, and this is similar to the C4 plants. The fixed CO2 is stored as malate or malic acid. During the daytime, malate or malic acid is then converted to allow the CO2 to become available. The available CO2 is fixed by RUBP in the Kelvin-Benson cycle. The figure on your screen is from the Plant Physiology Recommended Textbook, figure 8.13, and it shows an illustration of CAM metabolism. During the light, the stomata are closed, and the captured CO2, via the mechanism described in the dark, enables the process of photosynthesis to occur without the complication of losing water during the day. You may want to stop the video here and study this diagram in some detail so that you understand the processes involved in CAM metabolism. CAM plants have a 24-hour cycle, and this is divided into 4 stages. Stage 1 is the nocturnal phase. Stomata are open and CO2 is captured and stored in the vacuoles as maleate. Stage 2, early morning. Transient phase, allowing a shift in the metabolism to prepare for phase 3. Daytime is when phase 3 occurs. Stomata are closed, leaves are photosynthesizing. Stored malate is decarboxylated, resulting in high concentrations of carbon dioxide around rubisco, thus minimizing photorespiration and also water loss. And the final phase, phase 4, late afternoon, transient phase to prepare for the phase 1 activity of rubisco decreases while activity of PEPKs increases. So in summary, part 2 of this lecture on photosynthesis, the carbon reactions. During this, you should have learnt about the importance of RUBP. We've also introduced the concept of why oxygen is a bad thing in photosynthesis, and this results in a process called photorespiration. During part 2, you have learnt about C4 and CAM. These are different forms of metabolism and carbon fixation. They both involve complexities in their anatomy and have evolved other compounds like PAP to work efficiently. You are now able to compare C3, C4 and CAM plants. Thank you for your time.